This section will focus on protein synthesis for Chapter 3. Let's start with looking at the dogma of biology. So this is where we go from DNA to RNA to proteins. And so the nucleus is represented in this image here, and this is where we have our DNA. And so again, a gene is a length of DNA that codes for a specific protein. The gene on the DNA then is going to be transcribed, so with transcription, to mRNA, which can then leave the cell. Okay, and so this is considered to be genetic transcription. And so it goes from the nucleus into some type of, on a ribosome. Then the last step that we have is that the messenger RNA is then translated at the ribosome to assemble the proper amino acid sequence. And so this is what we term as genetic translation. These two steps of transcription and translation are going to be termed as genetic expression, where again we go from genes to what's made up as our functional protein. So let's start with transcription and then we'll go into translation. So transcription again is going from DNA to RNA. And so this starts within the nucleus of the cell. With DNA, here we have the DNA, and again, DNA has an alpha helical structure to it. We have some type of polymerase that comes in, and what that's going to do is that it's going to unwind some of that DNA. All right, and so within our DNA, there's always some region called a promoter region. And so these are areas of DNA that are not part of the gene, but will tell the enzyme where to begin. So the enzyme is this RNA polymerase, and a promoter region, say, is starting here within this nucleotide base. So then the purpose of RNA polymerase then is to break those hydrogen bonds that hold those base pairs together of DNA, and then they will assemble the correct RNA of mRNA within that nucleotide. And so here then we'll get complementary base pairing. Now again, one difference though is that RNA does not have thymine, rather it has uracil. So A now is going to bind with U, okay, for RNA, for that complementary base pairing. So the RNA nucleotide here with mRNA is now pairing with a DNA template. And so the assembly again is complementary. Thus, we have C's still binding with G's, A's are binding with T's for DNA, but now U's are binding with A's, again with the DNA. And so what this does then is that for the mRNA chain is it creates a precursor that's going to then later detach from the DNA template, where now that DNA then can start to reassemble again. And so only one of the free DNA strands is transcribed. Now, translation is the next process. So this is going from RNA to proteins. And so this is happening now on the ribosomes. And again, so we have our template that we made from the DNA double helix. And so now we had that transcription from DNA to mRNA. So you have to think about as it's like an interpreter for different languages, okay? So again, with the DNA, we had complementary base paired with our mRNA. However, the big difference here, let me circle in this example, is that A has been bound to U instead of to T, because now we're talking about RNA. So the mRNA attaches to a string of ribosomes to form a polyribosome. Okay, and so what we have then is for translation, now we're speaking a different language, okay? And what we have are groups of 3 bases on that messenger mRNA creates different codons, okay? And so, again, these are always by units of 3. The codons then are going to code for some type of amino acid, all right? And so there's always going to be a stop codon at the end. So here we'll have some type of stop codon, and then here some type of start codon. And so essentially the chain that's made based on those codons that essentially came from that mRNA that had been transcribed from the DNA is what makes then our chain of polypeptides, or forms our different amino acids. And so again, to make a polypeptide then, it ranges anywhere from 3 amino acids to 4,500 amino acids. And so the codons too only code for those 20 different amino acids that we find within the body. So tRNA, the transfer RNA, is also involved in the process of translation. And so with this, one end has this anticodon, which we see here, okay? And this is where the 3 nucleotides that will be complementary to the proper codon are found. The other end has the appropriate amino acid bonded to the amino acyl tRNA synthase enzyme. And what this does then is it brings the codon and it reads the mRNA sequence. This amino acid accepting end is what provides the appropriate amino acid then. Okay, so function of mRNA is to read DNA and make that into RNA. And then this transfer RNA is what will bring in the appropriate amino acid to make that correct polypeptide chain. So putting this all together then for the formation of polypeptides or proteins, what we have is that here in this picture, we have this pink is the ribosome, okay? Here we have our mRNA strand. Here we have our 3 nucleotide bases making the codon. And so the anticodons are going to be read then by the tRNA. And so the mRNA is going to move through the ribosome, and it moves very quickly, with the appropriate tRNA attaching to each codon. So what we see here blown up. The amino acids then attached to the tRNA are going to then form our peptide bonds. All right, and so here represents the formation of the growing polypeptide chain. The tRNA then will dissociate from the mRNA as it loses its amino acid. So here in this example, so tRNA no longer has its amino acid, so it unbinds, and then the next one comes in, okay? And so this will continue on until there's going to be some type of stop codon that will stop the entire assembly process. So interactions between then these amino acids of 1, 2, 3, 4, 5 here of this growing polypeptide chain will cause the difference in bending and then in folding in making the secondary and tertiary structures.